pure culture is crucial for accurate identification and further study of microorganisms, and streaking method is essential for obtaining a pure culture from a mixed population of microorganisms. There are several types of streaking methods used to isolate pure bacterial colonies on solid media. The main streaking types are quadrant streak method, T-streak method, radiant streak method, zigzag streak method, and continuous streak method. Today, we will learn how to perform radiant streaking method or technique, which is one of the most effective techniques to prepare a pure culture of bacteria. Welcome to this video. Let's get started. For streaking techniques, an inoculating loop is essential. It typically consists of a nichrome or platinum wire attached to a handle. In some cases, pre-sterilized disposable plastic loops are used. A Bunsen burner is also required for sterilizing the loop before and after use. To cultivate microbes, we need appropriate culture media plates, such as nutrient agar or selective media. Additionally, a mixed microbial culture plate is needed as the source from which we aim to isolate a pure culture. On the mixed culture plate, you can observe colonies of various microorganisms, differing in shape, size, and color, indicating the presence of multiple species. This is another type of plate that contains a single type of colony, which can also serve as a microbial source. Now, we will perform radiant streaking method. First of all, flame and burn the loop in the Bunsen burner until red hot. Let the loop cool for a few seconds. Gently touch and pick a single, well-isolated colony from the mixed microbial culture plate using a sterile loop. Prepare a smear using the bacterial cells collected on the loop on the edge of the media surface. Flame the loop in the Bunsen burner until red hot and let it cool for a few seconds. Starting from a distance edge, drag the sterile loop in a straight line passing over the smear toward the another edge of the plate. Repeat this step around the smear to create 4 to 5 evenly spaced streak lines. Repeat sterilization by flaming the loop. Allow to cool the loop without touching anything. Rotate the plate slightly. Streak from the edge of the initial streaks in a radial fashion, dragging the loop across the agar surface towards the edges of the plate. Repeat sterilization by flaming the loop. Allow to cool the loop without touching anything. Create horizontal streaks that intersect the radial streaks. This step further dilutes the inoculum, increasing the chances of obtaining isolated colonies. Close the lid of the Petri dish, invert the plate, and incubate it at 35 to 37 degrees Celsius for 24 to 48 hours to allow optimal bacterial growth. After incubation, carefully remove the plate from the incubator and prepare to observe the results. In the radiant streaking plate, you can see that where the initial inoculum was placed and streaked first, shows dense bacterial growth. This is expected, as the majority of the cells were deposited at the first streaked lines. Along these streak lines, the bacterial growth gradually becomes lighter. Toward the outer end of the streaks, isolated colonies are observed. This pattern demonstrates successful dilution of the bacterial load along the radiant lines, allowing for clear isolation of single colonies near the periphery of the plate.